castle where the digital dreams come alive. CESCG takes place from May 1 to 5, from visualization to the virtual space.
Okay,
Scientific sessions
welcome.
take their place.
My name is Norbert Vig, and I'm from the Slovak University of Technology in Bratislava. The title of my work is Multi-modal brain MRI registration using generative adversarial networks. What we work with actually are data about pituitary adenomas. Those are benign tumors with a prevalence of about 100 cases out of 100,000 people in the population. This kind of tumor, although being benign, can exert some pressure on the surrounding tissues, which means that it needs some surgical intervention in the worst cases. And the diagnosis is based on MRI scans, which can be either T1 weighted or T2 weighted. The difference between these scans can be seen in these pictures, where different kinds of tissues have different contrast, and the doctor then chooses which type of MRI scan is better for the proposed task. In most cases, they use both of these modalities, and also because of different tissues in these slices that can be examined, they also cannot annotate all the different tissue types. So this directly translates to what annotations we can get from these doctors. So for example, in the CET1 weighted scans, which are these blue ones, we have 4 classes, where in one class we can't distinguish between tumors and the cystic component. However, in the T2 weighted scans, we can distinguish between cystic components. However, we cannot distinguish between tumors and healthy glands. So to overcome this problem, we try to fuse these masks so that we gain better annotations. Eventually, we could use these fused masks in segmentation so that we can train a better neural network capable of segmenting in all these classes precisely. To do this, we actually have to register these images, but however, we cannot do it on the masks themselves, but we have to do it over the images of the actual MRI scans, because in reality we won't have always the masks, and if we want to segment it then it wouldn't make sense, because we couldn't do the registration, of course. So let's show some related works. First off, I would like to present the NiceGAN architecture. So this architecture is extension of the CycleGAN architecture. This one has 3 losses. One is the cycle loss, adversarial loss, and the reconstruction loss. And the other difference between CycleGANs and NiceGAN is that it reuses the encoder part of the network in the discriminator. This makes the training process more stable and easier. Moreover, there is a paper from Lee et al., and they try to synthesize MRI scans from scans of CT. And we can see that when they compare the different approaches, then the best performance was achieved by unit L1 plus L2, so using both regularization methods, and worst were using a PIX-to-PIX framework. Similarly good results were achieved by CycleGAN, which showed quite okay details in the core of the brain. Now let's continue with the solution that we created. The main dataset that we use comes from cooperation with a doctor from the Military University Hospital in Prague. It consists of 330 patients with pituitary adenomas. All of these patients were scanned in both modalities, so in T1 and T2 weighted scans. And the interesting part is that the T2 weighted scans are axial and the T1 weighted scans are coronal. Also, another point is that the spacing, although being quite okay in the field of the scan, in this slice we have about 1/4 to 1 millimeter of spacing. However, in the third dimension, the spacing gets significantly worse, where it can range from 2 to 5 millimeters. Also to mention, only a handful of these slices were annotated, so we don't have full annotations of these slices, which can also be another benefit of why we would want to fuse these masks. So as a first experiment, we tried to use a baseline image registration method. So we tried to find the transformation of coronal slices into the space of axial scans. The first step would be to interpolate the axial scans to create isotropic voxels. Then we would try to register these scans, and we would do that as a rigid transformation using the gradient descent optimization method. And to evaluate this, we would use the METS mutual information, since we are dealing with two modalities. So we can't compare the pixel intensities directly. As an output, we would get a two-channel three-dimensional data, as this is all happening in 3D. And as you can see from the results, we got some successful registration, but in most cases, this was unsuccessful, so we couldn't use this approach. Then we decided that we need to use our own registration algorithm. So this was caused because of the problems that I mentioned. So first off, the two modalities. So to solve this problem, we tried to use a GAN network, so a generative adversarial network. And by using this, we could rather use mean square error, so we could directly compare the intensities of the pixels. Also, another problem is that the moving image is approximately perpendicular to the fixed image. In our case, the moving image would be the coronal scans, and the fixed image would be the axial scans. Also, there are two more problems, that the spacing in fixed image is too large to interpolate, and the moving image sticks out of the fixed image. So what we do is we crop the moving image, then we interpolate it, and we extract a lot of subsets of slices with the same spacing as in the fixed image. Then we directly can register these. So this is a visualization of how this would work. So this rectangle here would be an overlay of the other modality after it was converted to this modality. So this is like, it would be perpendicular to these scans basically. So this is our pipeline. So the first step is to convert one of the modalities into the other using a nice GAN network. Then we would take the two scans, now in the same modality, and use image registration to find the transformation. So how do we do this? Is that we interpolate the C2T2 scans, then we generate these subsets that I mentioned before, and we identify the best subset of slices based on the lowest mean square error. From this, we get transformation parameters, and we apply these transformation parameters to the original pair of data. This way, we get a two-channel registered dataset, and we do the same transformation also for the masks. So to train GAN network, we also needed to use a dataset of scans from a dataset of registered coronal slices. So these slices come from 578 patients, which are all scanned in both of the modalities. So we can see two patients where both of the patients has scans in two modalities. These scans are registered together, so we know that these points correspond to each other, so we can directly compare them. Here I present the results of the GAN network. So we used the nice GAN architecture, and we converted the modalities. So basically, in the first column, we can see the original CET1 scan, and the second column is a generated CET1 scan. This scan was generated from the original T2 scan, and in the third column, we can see the difference between the original and the generated CET1 scan. So in a perfect scenario, this should be all white. That would mean that all the intensities are matching. However, in some regions, it's not exactly matching. The columns 4 to 6 are vice versa for T2 scans. So as you can see, it is probably easier to generate T1 scans than generating T2 scans. This can also be seen in our histogram of MSEs that we achieved over the dataset. So basically, the MSEs are lower when generating T1 scans. So in this slide, I'll also show the selection method of the best subset of slices. So we do this by selecting the index of the best subset of slices, which is the subset with the lowest MSE. So what we can directly compare is when we used simple normalization, the separability of the best result was quite easier. Like this was not the worst result between these two. So in this case, it would be still separable, but not that simply, kind of. We compared also with histogram matching, and the histogram matching also meant that it took significantly longer, and we didn't see any improvements, so we stick with simple normalization. In this slide, we can see 4 slices from one of the registered patients. So the light rectangle is the other modality that's overlaid over the axial scan. And as you can see that all the shapes of the brain are nicely aligned. When we evaluated over 77 of the patients from our data set, we identified that 4 of these were registered incorrectly. So we found that out by looking at them visually and seeing if there is a visible misalignment of the scans. And all of these 4 that were visibly misaligned were in the 6 of the worst results, which were all above this threshold, which we can declare as unreliable results, kind of. The success rate of our registration was 94.81%, which is quite better than what we had with the baseline approach. So to summarize, we trained the GAN network, we created the full processing pipeline for image registration with quite nice results. And for future work, we wanted to create a segmentation network that could validate our hypothesis that these fused masks could make the segmentation better. Thank you. This is all.
So here is now space for some questions from the audience. We have quite a few, please.
Thank you for the presentation. So I have multiple questions. One is quick. So what was the percentage for the baseline that you used? Because you said that the
I don't have it evaluated. It was probably run only over 10 patients, and out of these 10, it was maybe 7 were misaligned. So clearly, we didn't run it for all the patients, so I don't have it really evaluated, but we clearly saw that it wasn't performing up to our expectations.
Because it would be interesting to see like how much the different components, would change the result in terms of the percentage. If you already have like just the 4 patients that are misaligned, then you can probably calculate easily whether you are aligned or not, even for the previous cases. So it would be interesting to see, like for example, this GAN that we used, how much it helped. Because right now it's not clearly visible that what helps the
thank you. I'll definitely add it to my work so that I have it evaluated all.
And then, because then I have another question about, so for it's not clear to me that you are still using the gradient approach to find align them even in your approach, or is that just the baseline?
Yeah, it is also a simplified kind of gradient descent. So we have some kind of learning rate that adjusts the movement of the transformation in the way that it produces the best results. So it's a simplified version of gradient descent.
And then my question would be, so how do you initialize it? Because it seems interesting to me that the baseline works so badly, but I would assume that it's possibly because the initialization bad. Because if you are quite close, then the gradient descent with some good metric, as you are using like mutual information, should converge. So what are you using to initialize it?
Yeah, basically we can say that all of the slices are approximately, but not precisely, scanned in the same way. So if we put it in the middle, kind of, but not exactly, I have some average over all the patients, then if we choose this transformation, then it should be the closest.
And do you not have like some prior information on the transfer, like the position of the
No,
slices?
there is no guarantee, like we don't have ground truth of the transformation, unfortunately.
So even the scan doesn't contain like information about the...
No, we have only the spacing.
Okay, thanks.
Do you have an idea why the T1 images performed better than the T2 images?
Unfortunately, I don't have the answer for this. I would probably have to ask the doctor for this. But I suspect that it's different kind of tissues, which probably are simpler to identify in these scans. I don't know what you are trying to ask, but I'm not sure, really.
So maybe one last question. Okay, if it's not the case, then thank you very much, Norbert.
Thank you.
Swing to the beat where the pixels play, from rendering to VR. It's a pixel away.